particular the role of dopamine in value coding. So as many of you probably know, a central hypothesis in neuroscience is that dopamine signals a reward prediction error, the difference between the reward that you expected and the reward that you actually got. Now, it turns out that this error signal is extremely useful because you can use it to learn how valuable different features of the environment are. And you can also use it to learn which actions are more valuable to take in the long run. And which is interesting to us because we can measure serotonin. People have suggested that serotonin might carry the opposite signal to dopamine, coding reward prediction errors in a negative rather than a positive frame of reference. So to test these ideas in the human brain, Ken Kishida and Rosalind Moran asked patients in the surgical suite to play a sequential investment task in which they had to bet on stock price fluctuations in different historical markets. And here you might ask, why didn't you start with a classical conditioning task or a two-armed bandit or some other task that you might be more familiar with from the animal literature? And the answer is that we did try these different tasks, but it was too boring or too tiring for patients, so we switched to something more fun, more engaging. Now, because the markets are not fully predictable, you're going to experience outcomes that are better or worse than expected, given the market history. In other words, positive and negative reward prediction errors. And Ken and Rosalind found that on a large proportion of trials, dopamine and serotonin track these outcomes in the predicted manner. So dopamine increased for positive reward prediction errors, whereas serotonin increased for negative reward prediction errors. So we have found evidence that dopamine and serotonin support value coding. However, the task that I just showed you is a low-dimensional probe of the dopamine and serotonin systems. So we have a single valence axis ranging from punishment to reward. But recent work suggests that dopamine and serotonin play a much broader role to support a more diverse range of computations, if you like. So for example, we have evidence from human and animal studies that both systems are involved in action, with dopamine invigorating behavior and serotonin inhibiting behavior. And even in the value domain, dopamine and serotonin are not always opponent. So for example, serotonin neurons can be activated by reward prediction cues and by both positive and negative reward prediction errors. And I'll come back to what these kind of response profiles might mean. And we also have evidence that dopamine and serotonin track other dimensions than value and action, such as uncertainty about the environment or learning about associative relationships more generally. So what we did in the study I'm presenting today was to study dopamine and serotonin in the context of perceptual decision-making, where variables relating to sensory inference, choice formation, and behavioral control are all at play. And the idea is that if we use more complex or more multidimensional tasks, we can better tease apart or understand the contribution of dopamine and serotonin to thought and behavior. Now, an issue with many standard perceptual decision-making tasks is that they don't fully tease apart these component computations. So take the classic random dot motion task. Here you have to say whether a cloud of dots is moving left or right. And what we do as experimenters is that we make this decision easier or harder by changing the proportion of dots that move coherently relative to the number of dots that move randomly. But it can be difficult to interpret what a neural response means in this task. So let's say we find that when coherence goes up, dopamine goes up. But does dopamine track uncertainty about the sensory stimulus or the difficulty of the decision? Or does dopamine trigger the behavioral response, which can be prepared more quickly when coherence is high? So to overcome these limitations, we in this study, we used a modified version of the random dot motion task that I developed while I was a postdoc with Steve Fleming at UCL. So here's what we did. This is the task that we used both in the previous paper and in the current study. So first, what we do is that we use a continuous range of motion directions, not just left versus right. So here, it might be hard to see here, the angle of dot motion relative to the center is 10 degrees, but it could have been 90 degrees or any other direction around the circle. Second, we ask subjects to judge the motion direction relative to a variable reference direction, which only appears after the motion stimulus has terminated. So in this example, the correct response is to press the orange button. Motion is clockwise relative to the reference, but you cannot know this beforehand. So what we do is that, unlike the classic random dot motion task, we break the relationship between sensory inference and choice formation. You cannot prepare a decision until you've actually seen the reference. We also directly manipulated the demands on sensory inference and choice formation using a 2x2 design, varying sensory uncertainty and decision difficulty. So first, we varied the fraction of coherently moving dots in order to create trials characterized by different levels of sensory uncertainty. And the idea is that the lower the coherence, the higher the sensory uncertainty, the more uncertain you are about the actual direction of motion. And what we also did was to vary the distance between the motion direction and the reference direction in order to create trials characterized by different levels of decision difficulty. And here, the lower the distance, the harder the decision. So to illustrate this, what this factorial design means is that there will be trials where you felt fairly certain about the motion direction, but where the decision turned out to be hard. And trials where you felt uncertain about the motion direction, but where the decision turned out to be easy. Okay, so what does behavior look like? Do our manipulations work? So to answer this question, we're going to look at choice accuracy. And here I'm using the x-axis to indicate distance, low and high. And I'm going to use separate lines to indicate coherence, low and high. So here we have the data from the 5 patients in our study, 2 Parkinson's disease and 3 essential tremor patients. And indeed, coherence and distance have independent effects on performance. So choice accuracy is higher, A, when coherence is high, the red line is above the group, above the blue line, and B, when distance is high, both lines have a positive slope. And reassuringly, if we average across all trials, the patient's performance falls within the range of that shown by healthy controls, which I've indicated here by the gray bands. And finally, if we look at data from a pre-surgical visit, we see that the patient's performance is well matched before and during surgery, really demonstrating that we can run these kinds of tasks in the surgical suite, because naturally, having brain surgery is quite a different experience from performing the task on a laptop in a testing room. Okay, so now we know that our task works, so we're going to turn to our measurements of dopamine and serotonin. So I'm going to structure the results according to where we recorded from in the striatum. In primates, but not other species, the dorsal striatum is divided into the caudate and the putamen by an internal structure known as the internal capsule. And there's evidence to suggest that these 2 structures are part of distinct functional loops. A cognition loop passing largely through the caudate, receiving inputs from association areas and ultimately returning outputs to prefrontal cortex. And then action loop passing largely through the putamen, receiving inputs from sensory motor areas and ultimately returning outputs to premotor areas. I acknowledge that this functional description of these 2 areas is debated. So for example, by Susan Harper's work. So in our study, we recorded from the caudate in 4 patients and the putamen in 1 patient. And building on this framework, we're going to leverage this feature of our data to unpack the contributions of dopamine and serotonin to thought and behavior. And we're first going to look at the caudate data, and we're going to ask whether dopamine and serotonin track the cognitive variables probed by our task design. And we're leaving out one of the 4 caudate patients for reasons that will become apparent shortly. Now, the dopamine and serotonin responses may reflect a number of task variables. So take this example trial. Does the serotonin response reflect coherence, distance, their interaction, choice accuracy, or choice reaction time? Fortunately, because we have dissociated these variables, these task variables by design, we're able to answer this question, and we're going to start doing it start answering this question using a regression-based approach. And more specifically, we're going to do a sliding window regression. So here we have our design matrix or predictors, including our task variables for each trial. And we fit this design matrix to the dopamine and serotonin responses, sliding through time, running the regression model at each time point. Remember, we get measurements every 100 milliseconds, so 10 measurements per second. And what we get in return are encoding profiles, beta time series that quantify the relationship between our task variables and the neuromodulatory responses. So here the vertical lines indicate the onset of the motion stimulus and the onset of the reference. And the colored lines indicate the encoding profiles of beta time series. So pink is coherence, blue is distance, and green is the interaction. And overall, we don't see much happening in the dopamine response. By contrast, for serotonin, there's a strong negative effect of coherence, this task variable, during the presentation of the motion stimulus. So in other words, what this analysis shows is that serotonin in the caudate tracks our manipulation of sensory uncertainty. And here I was showing results at the group level, and importantly, we see this pattern not only at the group level, but also at the level of individual patients. So take the second patient, a PD patient as an example. So when coherence is low and sensory uncertainty is high, serotonin increases, and when coherence is low and when coherence is high and sensory uncertainty is low, serotonin decreases. So what about the fourth caudate patient? So here, both dopamine and serotonin appear to carry information about task variables at the very start of the trial. And in particular, we saw encoding of the interaction term, which really puzzled us, because at this point in time, you haven't seen the reference. So you don't know whether it's going to be low or high distance, if you like. So after we dug into the data and ruled out timing errors and so forth, all the different kinds of things you think of first, we then wondered whether dopamine and serotonin were sensitive to experience dependencies between trials. And in other words, we asked whether the two systems picked up on any coincidental structure in the task. So how could this happen? So our task turns the classic random.motion task into a discrete 2x2 design crossing coherence with distance. And this gives us 4 trial types. And each of these may be followed by another of the same 4 trial types. And the question is whether dopamine and serotonin picked up on coincidental structure in these transitions. The trial types were randomized, but the experience probability of transitioning between 2 trial types may seem non-random. So this is something that happens to us all the time. So imagine I flip a coin and it comes up heads every single time. So heads. Then I might wrongly think that I can predict the next outcome. So it's a random underlying process, but from my subjective point of view, it seems non-random. So the question is whether the two systems picked up on something similar in the task. So here we have an example of the probability of experiencing each trial type on the current trial, given that the trial type on the previous trial was defined by high coherence and low distance. And in this particular example, one of the trial type transitions is less likely than the others, and it would therefore be more surprising, if you like, from a statistical learning perspective. So interestingly, it turns out that both dopamine and serotonin track the experience task statistics. So in these two plots, the x-axis shows the transition probability of the current trial from the least to the most likely, and the y-axis shows the dopamine or the serotonin response in the current trial from the smallest to the largest. And in both cases, we see a strong correlation. And in case you're wondering, these relationships hold after we control for the marginal probability of each trial type. So it really seems that it has something to do with the transition between trial types. And it also holds after we control for sequential effects on choice accuracy. And I'll get back to what these results might mean or might tell us about dopamine and serotonin at the end of the talk. So we're now going to turn to our Putamen patient. And unlike the Caudate patients, we didn't see any encoding of the task variables. And this fits with the proposal that I mentioned earlier, that the division of the dorsal striatum into the Caudate and the Putamen reflects a cognition-action axis. But the question is whether we see any action-related effects in the Putamen data. Do dopamine and serotonin track the moment of choice, if you like? And the answer is yes. So here we have the dopamine and serotonin responses averaged across all trials. And the pink histogram indicates the distribution over choice reaction time from fast to slow. And as you can see, dopamine and serotonin track this distribution, with dopamine ramping up and serotonin ramping down. And in keeping with this idea of a cognition-action axis within dorsal striatum, we didn't see this response pattern in the Caudate recordings. So we think that these results suggest that dopamine and serotonin delivery to the Putamen may trigger the execution of a choice. And if this is what happens, then we would expect these changes to happen earlier for faster choices. So to test this hypothesis, we're going to take our trials and using terciles over reaction time, we're going to group them into fast, medium, and slow. And the prediction is that the changes in dopamine and serotonin happen the earliest on the fast trials and the latest on the slow trials. And this is what we see. So here we have the average dopamine and serotonin responses for our three time bins. And as you can see, dopamine peaks early on fast trials indicated by pink and late on slow trials indicated by blue. And similarly, serotonin drops early on fast trials and late on slow trials. And I think it's even more apparent that these responses are tied to action when we realign to the moment at which a choice is made. So what we see here is that regardless of the time taken to make a choice, dopamine ramps up, where serotonin ramps down immediately before a choice is made. Here the vertical line indicates the moment at which the patient presses a button to indicate the choice. So to use a car metaphor, it looks like dopamine plays the role of the accelerator, creating movement, where serotonin plays the role of the handbrake, holding movement and then allowing it to be released. So just to remind you here, zero doesn't mean that there's zero serotonin. It just means it's at some baseline level and then it drops. Okay, so what I'm going to do now is to zoom out and sort of look at the bigger picture and contextualize the results that I just showed you. So we found evidence that serotonin tracks uncertainty about a short-lived sensory stimulus, and I don't think this particular result has been seen before in any species. There are animal studies that have linked serotonin to uncertainty on another class of task, reward-based decision-making, where animals have to learn from experience which choice, in this case left or right, is most likely to produce a reward. For example, serotonin neurons track not only signed reward prediction errors, whether an outcome was better or worse than expected, but also unsigned reward prediction errors. And these tell you how surprising an outcome is, irrespective of whether it was better or worse than expected. And this you can think of this as an uncertainty signal. So the fact that we find this computational motif across domains and across timescales suggests that serotonin may encode a generalized uncertainty signal. So why is this a useful signal for the system? Well, at fast timescales, uncertainty is an important signal for behavioral control. So for example, if you're driving through the rain and you don't really feel like you can see what's in front of you, then it might make sense to slow down. And at slower timescales, uncertainty is an important signal for learning. So take this scenario. So the left option has led to reward in the past, but now you take the left option and you get nothing, a big surprise. So this tells you that you can no longer be certain about the task rules. The world might have changed, and perhaps you should update your model of the world accordingly. And we do have causal evidence that serotonin drives this kind of plasticity. So for example, if you stimulate serotonin neurons on particular trials, it turns out that those trials have a bigger influence on an animal's future behavior. And I also think that this is one of the cases where we can speak to clinical conditions, although admittedly this is more speculative. So take SSRIs, which increase serotonin levels in the brain. So SSRIs have been shown to be a common and sometimes very effective treatment for mental health conditions such as depression and anxiety. But perhaps the reason why SSRIs can help break rigid thoughts and behaviors is not because of any direct effects on mood, if you like, but because of general uncertainty-induced plasticity. So a surprising aspect of our data was that dopamine, at least at the group level, didn't really track our task variables. So why is this surprising? Well, as shown by Armin Lack and colleagues, we know that dopamine neurons track motion coherence on the random dot motion task. So the higher the coherence, the higher the firing rate in midbrain dopamine neurons. So why didn't we see this? Well, it might have to do with the disease state of our patients, although I was shown data from both PD and ET patients. It might have to do with our factorial design, which breaks the direct link between motion coherence and choice accuracy or choice reaction time. Or perhaps it's because we didn't provide trial-by-trial feedback or reward signal, if you like. But if you look into the supplementary materials of our paper, you see that dopamine levels do fluctuate across time. We just can't make sense of them or tell some simple story about them using our factorial design. So again, this is speculative, but perhaps the reason is that dopamine reflects more specialized computations. For example, coding for particular parts of the stimulus display or particular stimulus action pairings. And we do have some reason to believe that this might be the case. So in this recent paper, Ben Engelhardt and Ilana Witten showed that distinct clusters of dopamine neurons encoded distinct aspects of cognition and behavior. So in our case, perhaps we shouldn't expect to see anything clear-cut when we average across physically very different instantiations of a particular task condition, and especially not at the group level where we also average across different sites of dopamine release. So moving on, in our Putamen patient, we found that both dopamine and serotonin tracked the moment of choice, suggesting that they're involved in triggering action. And both of these results fit with recent animal work. So, for example, optogenetic stimulation of rat dopamine neurons increases the frequency and the vigor of self-paced movements, consistent with the role in dopamine invigorating behavior. And conversely, optogenetic stimulation of serotonin neurons increases the amount of time that rats are willing to wait for an uncertain reward, consistent with the role of serotonin in inhibiting behavior. But also want to highlight a striking similarity between these neuromodulatory responses and electrophysiological correlates of perceptual decision-making in humans and non-human primates. So here I'm showing human EEG data, and in particular, an ERP known as the central parietal positivity. But you can see the same qualitative pattern in single cell recordings from monkey LIP, for example. So just like in our data, the neural responses separate by reaction time, fast, medium, and slow, when we time lock to the onset of the stimulus, but not when we lock to the moment of choice. And one interpretation of these data is that the underlying neural areas support the accumulation of sensory evidence towards a choice bound as conceptualized by the drift diffusion model or other sequential sampling models. So it might be that these changes in dopamine and serotonin levels reflect an evolving decision variable rather than the triggering of an action once evidence accumulation has been completed. We'd have to do more sophisticated experiments, really dissociating evidence accumulation from moment of choice in order to get at this question. So finally, a surprising aspect of our data, which we hadn't set out to test, was that dopamine and serotonin tracked experienced task statistics in one of our patients. So our task doesn't allow us to isolate the underlying computational mechanism. So the responses could reflect some cognitive construct that is modulated by task statistics. For example, I might hope dread particular trial types, given what I've experienced this far. Or they could indicate a more direct role for dopamine and learning, for dopamine and serotonin in building, in learning a statistical model of the outside world. So this statistical learning interpretation fits with recent work from Jeff Schoenbaum's lab on dopamine and sensory prediction errors. So this is a sort of a human example of the rat study that they ran. But let's say I prefer Coke and Fanta by an equal amount. In other words, they have the same value. And what Jeff Schoenbaum's lab has shown is that dopamine neurons track not only changes in value, for example, getting more Coke than I expected, but also changes in identity, for example, getting Fanta when I was expecting Coke. In other words, a change in the sensory features of a reward, but not a change in value. And these results are exciting because they suggest that dopamine also supports the learning of an associative model of the environment, helping us learn the statistical relationship not only between states and rewards, but also between states themselves. And here I use state in an abstract sense. States could be stops on the tube or different objects in a visual scene. And if you want to learn more about this idea, then I recommend reading these papers, and in particular, this paper by Thomas Ackham and Mark Walton from these guys here in Oxford. Now, one issue with this so-called trans-reinforcer task, and thereby the evidence for sensory prediction errors, is that value and identity are yoked. We don't know whether sensory prediction errors are limited to features of a reward, in this case, the flavor of a reward. We also can't say for sure that we're looking at identity and not value. So for example, while I prefer Fanta and Coke on average, by an equal amount on average, I might feel like a Fanta if I've been drinking Coke all day and vice versa. So there might be some time-varying, consumption-dependent preference changes at play. So what we're doing now is that because we think this idea is really exciting, is that we're planning to run a study where we fully separate sensory statistics from reward. And the basic idea is to have a task where the start state fully predicts whether you're going to get a reward, but where the probability of different sensory experiences along the way to the reward depends on the start state. So what do I mean? So let's say I get into my car to drive to Starbucks, which is at this moment like a realistic scenario. So I could do this in the morning or in the evening. And in both of these cases, I'm going to get my coffee, my reward. But the sensory statistics of this trip, the probability of what I see out of the window, is going to depend on the start state, the time of day I set out. So in the morning, I'm more likely to see the cow, but in the evening, I'm more likely to see the owl. And the question is whether dopamine and perhaps serotonin track violations of these sensory expectations, even though these violations are entirely non-consequential for reward. We're currently piloting this task, and hopefully I can come back to tell you what we find. So in the last couple of minutes, I'm just going to also highlight some other potentials for studying fast neuromodulation in the human brain. So today I've talked about dopamine and serotonin, but when you adopt this statistical machine learning approach to analyte inference, to signal inference, you realize that there's no reason to think that you couldn't study other neuromodulators. So traditionally, the idea is that it's hard to separate these neuromodulators when you look at voltammetry data by eye, or when you look at the first or second principal component. But perhaps we can disambiguate them if we throw lots of training data at our model and then just ask in an objective manner, can our model recover fluctuations in these different neurochemicals at the same time? So here I just wanted to flag some evidence that we can perhaps also look at noradrenaline. So while, as far as I know, that there's not a lot of noradrenaline in the striatum, there are no projections to the striatum, we can also do recordings in other parts of the brain, which I'll get to in a second as well. And here I'm just showing some proof of principle data from the wet lab where we have a model that's trained on a mixture of different neurochemicals, and then we apply to withheld training data. So we have 5-HIAA, which is a metabolite of serotonin, and then we have noradrenaline and dopamine. And as you can see, the model, which are the dots, sort of follows fluctuations in the different neurochemicals in this test data. So it seems that perhaps we can also look at other neurochemicals at these fast timescales. And as I alluded to earlier, it's this far we've used carbon fiber electrodes, but there's no reason to think that you couldn't get measurements of other kinds of electrode. They might not be as good as of the carbon fiber electrode, but we should definitely explore the potential of using other platforms for obtaining these measurements. And here I'm showing proof of principle data using a depth electrode that is applied for monitoring of seizures in an epilepsy patient. So what you do sometimes in severe cases of epilepsy is that you implant depth electrodes for monitoring for a period of time in order to localize where the seizures originate. And these are in place for let's say a week, which means that you could perhaps study neurochemical fluctuations on different tasks or at different times of day, or on the same task at different days, on different days. And here I'm just showing, again, proof of principle data that using this electrode, but in a wet lab environment, that we can get good dopamine, serotonin, in this case, the serotonin metabolite predictions of an electrode like this. So it really opens up for studying neuromodulation, not just in the structures that are sort of accessible as you follow the surgical trajectory through the common targets for DBS surgery in PD and ET, but you can also look at neuromodulation in other areas as probed by depth electrodes in epilepsy monitoring. So to summarize, I hope to have convinced you that dopamine and serotonin support a diverse range of computations and not just value coding as we might traditionally have thought. And with that, I thank you for your attention. Thanks.
Okay, thanks very much, Dan.
Thanks, Matthew.
So are there any questions? One thing that Nima told me to tell you was that we're recording the lecture. So I think I'm supposed to say that before you ask. So while people are just thinking of some questions, one of the things that I was wondering about, Dan, is that some of the one of the things that makes me wonder is whether or not some of the hypotheses about what different neuromodulators are doing are partly based on what the investigators are interested in, and then they investigate those things with a particular neuromodulatory system that they're interested in, but we never really look at some parallel neuromodulatory system. So it sort of suggests that some of the specificity that we had thought existed in some of these neuromodulatory systems might not be quite as substantial as we thought before. Whereas with this approach, because you can measure several things at the same time, you're able to actually compare and contrast serotonin and dopamine directly. Is that?
Yeah,
comments or thoughts about that?
No, I think that's a good point, because, so for example, when you see these kind of opponent effects, it suggests to you that there is indeed some kind of underlying construct that these systems are operating on, and they're signaling something about that underlying construct. And I also think you're right that, imagine we had not seen this result, I would say, oh, and not this one, I would say, oh, these systems are doing action. Look, they're doing action.
Yeah.
But over here, they're doing something slightly different, and it might be hard to reconcile this response pattern with the serotonin response pattern over here. So is there some unifying explanation of this? There might be, but they might also be doing different things, right? Because they're different cell types, they're different receptor types, they're receptors in different neural areas that specialize in different kinds of things. But I do think you're right that a lot of the focus on value comes from an interest in value, and also because it's obviously a very general, potentially general explanation.
So there's a question from Pip.
So, it's actually Pip's husband, Lawrence, here. Hi, Dan. Thanks for that. It's a great talk. So I just wanted to clarify the distinction between the sensory uncertainty and the response encoding in serotonin. So it seemed like those two could kind of conceptually be quite closely related to one another. Like when your uncertainty is higher, you might take longer to respond. And so is the distinction really just that one is found in the putamen and the other is found in the caudate, or did you try and look for both of those signals, or kind of regress one out? And
no, I think that's a good point. So just to like add further to what Lawrence said is that. So here the idea is that serotonin is inhibiting action, and then when it drops, an action is made. And you could say that that's consistent with the pattern over here, because on average, you are going to respond faster on high coherence trials than low coherence trials. So, but I do think that, yes, it could reflect sort of two sides of the same coin, but also think that there's perhaps more to this caudate Putamen distinction, because over here we find this result when controlling for using our sliding window regression, when we control for reaction time, when we control for distance, when we control for the interaction between coherence and distance, which also could contribute to when you're going to make a choice. But it might not be saying hold on for action. It might be saying hold on for some other cognitive process, such as stay on task or something like that. But yeah, again, like going back to Matthew's point, there might be some higher level construct that could reconcile these findings, and I think it's a good question.
And just following on from that, so is the organization of, I don't really know that much about the organization of serotonergic projections to those two parts of the striatum. Are they organized in discrete sets of sets of neurons in the same way that they are for the dopamine system? Or
well, that's a good point, and I know definitely much less about that than I do for the dopamine system. And you're absolutely right that so the caudate is more similar to what we might think of as dorsomedial striatum in the rat brain, whereas the putamen is more similar to dorsolateral striatum in the rat brain, which we also think of as having these cognition-action functions. But that's obviously true when we look at activity in these areas and different tasks, and it also seems to be true for the projections to those areas, that there might be some stratification down in the midbrain dopamine neurons. I know less about it for serotonin, and I think perhaps one reason why a more generalized explanation that could reconcile this finding might be preferable is that while there are many different, I think there are more different receptor types for serotonin, but I think that there are fewer serotonin neurons. So it could suggest that it's doing something more generalized or global.
I think there's another question from Marianne Noonan.
Hi, Dan. Thank you. I think that's super cool stuff. These were patients, right? And patients with Parkinson's, yeah?
So some of them have Parkinson's disease and some have essential tremor.
Yeah. Okay. Did you give them any questionnaires related to mental health? Because I'm wondering how typical these patient groups are, particularly with serotonin, particularly with often these conditions can be comorbid with things like anxiety and depression. Did you measure that at all?
Yeah, so we have access to the medical history, and people are working on that. I just want, so in this particular case, it's quite hard to make these kind of stratified inferences, because here I have a slide, but I'm not going to move to it, where I show the medication profile for each of the 5 patients. You can see they're all on different kinds of medication. Some are on SSI, some are not. So, and of course, all these different kinds of things could affect the responses. So here the idea is more that, we see a similar thing across quite different people with different medical profiles. But your point is really relevant, and I know that so Ken has a bigger paper where he looked at with the sequential investment task, where there's, I think, 20 to 22 patients where he does stratify. So this is not published work, but where he stratifies it according to whether people are on SSIs or not, and it does look like that there are different responses in these 2 groups. So obviously that plays a role, and I definitely think as you acquire more and more data, maybe you can't relate medical history to particular responses, but you could perhaps look at more general properties of the system, such as how they respond to stimuli or how they're involved in action and so forth.
Yeah, really interesting. As ever, always more data.
Yeah, exactly.
It's super cool work. Thank you.
Thank you.
Any more questions? Okay. Well, in that case, seeing as we're exactly at 2 o'clock, so we'll draw things to a close and say thanks very much, Dan, and thanks. We'll see if we can give you a round of applause.
Good luck.
Okay, well, thanks very much.
Thank you, and thanks for the questions.